Howdy. So this project is based on some work that was done a couple of years ago, 2 years ago at Harvard. These guys figured out a way to get a virus to evolve a special protein, sort of your own design. And it's called PACE, which is called phage-assisted continuous evolution. And the phage it refers to is just a bacterial virus, and that they've worked it out so that you take a gene out of the bacterial virus that makes it infective, and you remove that gene from it, and you put that in E. coli, and you make it so that the E. coli, if you get a certain protein binding, a certain protein, the one you're trying to evolve, that the E. coli will then produce that protein for the virus. And so basically, the way we're putting evolution to work here is we have an error-prone DNA polymerase. It makes lots of errors when it copies DNA, and that gives us variation. Every time the virus goes through a generation, we've got this mutagen DNA polymerase that makes lots of mistakes. So every generation of virus, you get 1 billion different copies of the virus with 1 or 2 mutations somewhere in its DNA. Then, that virus, as it goes into the E. coli will make the proteins from the viral sequence, and if it makes a protein that binds with the target that we have set up, it will produce the P3 protein, and therefore that virus gets rewarded by making it, its progeny, its children, will become more infective. So basically there's a diagram of how it goes. We don't really need to get too much of the detail there, but the basic idea is that we add these 2 extra chromosomes, MP and AP, to the E. coli. One to cause the mutagenesis, the error-producing DNA polymerase, and the AP is the feedback mechanism where we reward the virus if it produces a protein that does the binding that we want. My project is phage-assisted continuous evolution on a budget. So the Harvard people describe in a very nice supplement to their paper, they describe how you can build this apparatus for about $25,000. And what I'm doing is using the Arduini actually, for $10, and scrap exchange parts to build a thing for around now about $400 is what I'm getting to. So anyway, so this is how this, you put evolution to work here, and I'll show you, there's the apparatus basically. And if you turn around, you can see the actual apparatus here. And what we've got is a system where I've got a stirring platform mixing the E. coli hatchery, and then the small bottles or the lagoons where the viral evolution is going on. And if we look back over here, I'll show you how I'm measuring the levels in the containers. Let me get this. I started measuring level with pressure sensors, and I had to build so many of them. And then I figured out that Open Computer Vision, OpenCV, is a open source system. You can just get an image and run the Huff transform on it, the Huff image detection, to look for the horizontal features, and you can actually read out the levels in the containers. So, so now I've got a really cheap $20 webcam solution to measuring the levels inside the containers. And I'm putting light in the mixing platform that shines up and causes the level of the water to glow. And when you close this whole thing up, you get the control the light level so that it just doesn't get any reflection over here. Anyway, so you got one styrofoam box here, a heating blanket that's run by the Arduino, and that keeps the E. coli growing at 87 degrees Fahrenheit. And then we got the webcam, which is letting us see what the levels are. And then I have 8 peristaltic pumps that are pumping the fluids into that container. And if you come around here, you'll see the overall result is something we call the PACE capsule. Okay. And so you push the 2 styrofoam boxes together, and you've got your thermally controlled environment. You pump liquids in. It's growing, and it's continuous evolution. That part of the name means you can leave it now for a week, and the virus just goes through one generation after another to get better and better binding at the target that's in the E. coli. And so the 2 real breakthroughs that made it all cheap is using the Arduini to drive the peristaltic pumps and to measure the turbidity of the E. coli culture with a sensor, photosensor, and then using OpenCV to grab the chemical levels. So I can't take credit for the basic idea. Again, Asphalt, Carlson, and Lou at Harvard are the guys who came up with the whole paste concept. But I can do it for on the order of $400 or $500 instead of $25,000. And a friend of mine at UNC who wants to use paste to evolve a protein is very happy because he wants to build 4 of these apparatuses, and now we're talking about $2,000 instead of $100,000. So I get my kicks building stuff from the scrap exchange out of cheap parts or discards, and I'm the engineer at the Museum of Life and Science, and this is what I do in my spare time. I've been interested in molecular biology for all of my life and been on the fringes of it and do little projects like this. So that's evolution as a tool, or open source evolution as I like to call it.